morning, good afternoon, and good evening, everyone, from wherever you're joining us. And welcome to today's cutting edge conversation titled Artificial Intelligence in Preclinical Research, Pathology, Biospecimens, and Model Selection. I'm Liam Sanyo from the events team at scientist.com, and I'll be your host today. Joining us are three experts: Meredith Osborne, Associate Director of Product Management at Discovery Life Sciences. We have Daryl Waggett, Director of Biologics and Data Products at BioIVT, and Luke Jervis, Senior Software Engineer at Certus Oncology. Today, they will share practical applications of AI, from quantitative image analysis to leveraging curated data and optimizing tumor model selection. All right, let's get into it. So at this point, artificial intelligence is being applied across every stage of biomedical research. But today we're narrowing in on its role in preclinical workflows. So this is an area where researchers face some serious bottlenecks, but not always because of a lack of data, but because sometimes the data is hard to access or difficult to interpret at scale, or not easily integrated with the context needed to make it useful. And the pressure to improve reproducibility and translatability, reduce costs, and bring therapies to patients faster is only increasing, while the complexity and volume of data can really become a barrier. And I think that's really where AI will continue to prove its value, by really helping researchers to work more efficiently or see patterns that they might have missed, reduced subjectivity, integrate more types of data, ask more precise questions, and surface insights that support better study design and model selection, all of which really contribute to more predictive and translatable research. And I think we'll also really start to see AI shift towards being more of a back-end analytics tool to something more integrative and interactive. And with that, I'm very pleased to welcome our first speaker, Meredith Osborne. So Meredith, thanks so much for joining us today. And the floor is yours whenever you're ready.
Thanks, Liam, for the introduction. So my portion of today's webinar is titled Beyond Human Eyes, How AI Turned Routine H&Es from Qualitative to Quantitative Insights. As Liam said, my name is Meredith Osborne. I'm the Associate Director of Product Management at Discovery Life Sciences. Here at Discovery, we are both a biospecimens provider, so we have a commercial biobank for samples available for research use, as well as a specialty lab services provider. So throughout my talk, there are going to be a few kind of bigger themes that are applicable to whatever type of AI is relevant to your work or that you're looking at implementing. And the first big umbrella theme is fit for purpose AI suite. And what I'm getting at here is that it is best to think of AI as lots of tools in your toolbox that each have a specific job. Different data inputs that they need, and then different data that they give you at the end. So AI is not, kind of one giant, super intelligent, multi-purpose tool that can handle everything. It's a toolbox with lots of tools, and it's all about picking the right tool for the job that you're trying to get done today that can accelerate your project. Within kind of that broad theme of thinking of AI as a suite of tools that are each fit for purpose, I'm going to cover an example of AI doing things that humans can do, an example of AI doing things that humans don't do, and lastly, we'll touch on an example of AI doing things that humans genuinely can't do. And all of my examples are going to be featured around AI analysis of H&E images. H&E images are a very standard microscope slide that's used by pathology centers globally, but they're fairly old. They were first introduced in 1877. Then in the 1920s, the American College of Surgeons mandated that all tissue removed from patients during surgery or biopsy be examined in a lab, which means H&Es be made, and reports be written. Well, so then those reports then got standardized. 1946 was the founding of the College of American Pathology for the purpose of standardizing these pathology reports, which are just written descriptions of what people see when they examine these H&E microscope slides. So the reason that H&E slides are still in use, still remain standard of care today, even though they're in the world of medicine, such an old technology, is that they are such a rich source of spatial and morphologic data. for research, as well as for diagnosis in a clinical care setting. But historically, these are pictures, and even our reports are written text reports. They lack structure and they lack scale. And that's where AI can come in and help us, continue to innovate and move forward. So our first example is AI doing something that humans can do, and that something is tumor percent quantification. So on the left, when a human looks at an H&E image, in this case, a very highly trained human with the title pathologist, pathologists are able to see where the tumor is. So in this particular one, most of the tumor is circled there on kind of the right side, and there's a small focus of tumor on the left. And if you ask a pathologist to say, hey, what percentage of the sample is tumor, you'll generally get answers between 5 and 15% for tumor like this one. AI, on the other hand, can do that same job. So using an AI called Tumor Detect from one of our partners, Path AI, it also identifies where the tumor is. In this case, it shows up as orange and pink. Orange is the cancer stroma, pink is the cancer epithelium. But same findings, right? Most of the tumor is there on the right, and there's that small focus over on the left. But unlike humans, when AI calculates the percentage, it's very precise. For this particular sample, it is exactly 8.65% tumor. And this variability in human tumor percents is something that we've seen internally for the decades that we've been in biobanking, right? FFP is one of the many sample types Discovery has in our commercial biobank. And what I'm really trying to show with this graph is that even though the human pathologists are giving us a number, it's still fundamentally a qualitative observation. So, like this third sample is a good example, right, of what we wish was common. The pathologist with the blue score said 95%. The one in orange also said 95. The one in green said 100, right? So, high level of concordance. The next sample over, the fourth one, the pathologist in blue rated it 40% tumor, the one in orange rated it 90, and the one in green rated it 65. Right, so a pretty significant amount of disagreement, not in the fact that there is tumor, right? The diagnosis, the presence of tumor, all the pathologists are in 100% agreement there. But just when you ask them to put a number to, these irregular shapes, what portion of the irregularly shaped tissue is comprised of the irregularly shaped tumor, you get some wide variability. And that's where AI can come in and give us this highly reproducible, very precise, truly quantitative tumor percent. And so here on the right, we see 6 different samples were run through Tumor Detect twice. And as expected, the computer gives us the exact same tumor percent every time. So our second example, AI coming in and doing something that humans don't do. And what I mean here is like technically humans can, but we don't because it's not scalable. So this example is enumerating the presence of tumor-infiltrating lymphocytes. So these are the body's white blood cells in your immune system that are present in that tumor microenvironment. These are really important cells when you're doing immuno-oncology research. That is, kind of big picture, all about how can we arm the immune system to fight the cancer for us. So, human on the left, again, very specially highly trained humans, pathologists, are able to look at this picture, zoom in, see cells. They can recognize what white blood cells look like. They can recognize where tumor is and if those white cells are present in the tumor. And generally, the readout then that the humans give us is whether there is a high presence, a medium presence, or a low presence of tumor infiltrating lymphocytes in this particular biosample. And now for AI, we've switched tools in our toolbox. Last time we were talking about Tumor Detect. Now we're talking about an AI called Discover AI that was co-developed with Discovery Life Sciences and our partner Path AI. And like Tumor Detect, Discover AI breaks the image into areas, right? Where is the cancer epithelium in pink, the cancer stroma in orange? And then, like the human, the AI can zoom in, identify every cell in the image, and classify it. Is it a cancer cell, a fibroblast, a plasma cell, a lymphocyte, a macrophage? And since we're talking about tumor infiltrating lymphocytes, it's really those lymphocytes right in the tumor area. And unlike the human who gives us these qualitative high, medium, low assessments, Discover AI gives us quantitative spreadsheets. So here we see just a small snippet of example data. The area of cancer is the second column there in millimeters squared. And then that third column is really our TILs, tumor infiltrating lymphocytes. How many total lymphocytes are there in the cancer stroma? On the low end in this example set, we see 2,281 lymphocytes, and on the high end, we see 18,822. Then once you have these cell counts and areas, you can also do, various ratios and densities, such as the last example, the final column ratio of lymphocytes to fibroblasts in the tumor. And it's not that humans can't do this. Humans are totally capable, after being trained and becoming pathologists, to recognize lymphocytes, see which ones are present in the tumor. But counting to 18,822 is an unrealistic ask. Even on the low end, counting to 2,281, also an unrealistic ask. So humans can do this, but they don't because it's not scalable. And then our last example that I'm going to end with before handing the floor over to our other speakers is some AI coming in and doing something that humans genuinely can't do. And like the other two examples, we're still starting with that H&E microscope slide, that's standard of care created every surgery everywhere in the world. But now we're adding in a second data source. We're pairing that image with treatment and outcome data. After the surgery happened, what drugs was the patient given, and did they work or not? And when you pull out the right tools from your AI toolbox and you give that AI both of these data sets, you're now able to generate something that humans can't, which is truly novel predictive biomarkers from that H&E image. As an example of these, PathAI presented at AACR in 2023 a case study where they did this, and the novel biomarker found was tied to the ratio of immune cells to fibroblasts in the cancer stroma. And for the drugs being studied, that biomarker was predictive of both progression-free survival and predictive of overall survival, which of course, overall survival is kind of our main end goal as drug developers and clinical trial design. Overall survival is data you hope to be able to get and the endpoints that you most want to improve. And Discovery can come along, partner, and further this type of work with the portion of our biobank that has up to 2 years of patient follow-up data. So this is us, after our biospecimen was collected, continuing to follow that patient for up to 2 years and seeing what treatment did they get? Did it work? What is the clinical data that backs up the claim that the patient responded or didn't respond, whether that's blood biomarkers or imaging data, and kind of capturing all of that so that this type of novel biomarker prediction can continue. So, in summary, uh, AI allows us to take H&Es, which have been for over a century a rich source of qualitative data that's so valuable it remains in use globally today. But instead of leaving it qualitative, just kind of like paragraphs of text, AI allows us to take that and make it truly quantitative, numbers in a spreadsheet that are very exact, very precise, very reproducible, and very ready for mathematical analysis. And within that, these were all examples of, our big theme. Think of AI as a suite of tools that are each fit for a particular purpose. So it's all about choosing the right tool from your AI toolbox. And I gave an example of AI coming in and doing something that humans can do. We saw that picture of a slide with 8.65% tumor. AI doing something that humans don't do, counting the number of tumor infiltrating lymphocytes, which in this picture is 18,822 on that full slide. And then lastly, AI coming in and doing something that humans can't do, which is identifying novel predictive biomarkers in the H&E image. And the example we gave was a biomarker tied to the ratio of immune cells to fibroblasts in the cancer stroma, predictive of both progression-free survival and overall survival. So if you have any questions specifically after the webinar, feel free to reach out to us at info@dls.com, and stay tuned. We'll be releasing a white paper in the next few weeks that goes into these same topics at a little more detail. But for now, Liam, back to you.
And with that, I'll invite all of the speakers back to the audio and video line for the Q&A. So I think, Meredith, we'll start with a question for you here. Why is the tumor percent important?
Yeah, thanks, Liam. So tumor percent in a clinical setting, like when we're talking about patient care, actually is not important. When pathologists are writing path reports, tumor percent is not one of the things they list. But in a research setting, thinking about research that happens, preclinical, prior to clinical trials, it's a key data point. So it helps researchers select the right samples that are suitable for their projects. And as an example, let's say you're using FFP tissue for bulk genomics. Uh, Variant allele frequency is a real number that you need to contend with. So let's say, not that you know this yet, but there is some mutation that's present in 5% of the tumor cells. Well, if only half of your sample is tumor, that 5% is now 2.5% of the total, which may fall below, kind of your minimum detectable threshold.
Fantastic. Great answer. All right. Next question here will be for Luke. How are the drug response predictions generated, and which drugs are included? And can I create predictions for my own compounds?
Yeah, thanks, Liam. So we have a patented tool called Certus AI, and it's a machine learning model that's trained on hundreds of thousands of in vitro and in vivo experiments. Um, Basically, the features of the model, so what it's trained with, are gene expression biomarkers. So these are specific genes that we've used a biomarker selection tool model to generate for each cancer type. And then on the other side, you have the molecular structure, the SMILE string of the drug itself. So we can learn on both ends of that pairing. And we then train the model and then use that trained model to predict response on novel drugs and new biological subjects. The assistant itself has a pre-computed library. So we've gone ahead and run this machine learning model on every FDA-approved small molecule drug against every model in the Certus catalog, along with every model in the CCLE catalog. And those are available to the assistant to pull from. And you could see on some of the examples I showed in the presentation that it can create violin plots to compare cohorts based on these drug prediction responses. Now, currently with the assistant, you're not able to input your own drug or cancer model, but that is something that we're working towards. And it's, I think, something that's going to be very relevant and very interesting for people to get their own predictions in there and see where they stack up and compare to all the other predictions that we already have pre-computed. So that's something we're working on, and we're hoping to have something prepared in the first half of 2026.
Fantastic. Thanks a lot, Luke. All right, Daryl, over to one for you. It's more of a subjective question, I think, but what aspects of the drug discovery pathway do you think are best suited for AI?
Thanks, Liam. Well, I think the simple answer is everything. The entire drug discovery pathway will be impacted by AI over time. I don't think that's very controversial. We're all aware there's a significant amount of momentum around AI for target discovery, lead prioritization, molecule design. I mean, you can't, not read a sort of life science news article without having discussion of those topics. But what I think is uncertain is around the white space for disruption because of so many different players going at it. And specifically around where the opportunities are for adoption, implementation, deployment. I think that where we see a lot of sort of not necessarily failures, but sort of a little bit of headwind is, the technology is great, the models are developed, but when it actually comes to deployment, implementation, adoption, we see that there's some sort of challenges getting it across the line and getting it sort of working. Where are those barriers? I think a lot of them are not technology. They're focused on the regulatory constraints. They're focused on the cultural constraints. People like doing things in certain ways, especially in biopharma. And so there's a passive resistance often to that adoption. Where I think we'll get the initial traction in adoption, I mean, that's already happening across the board, is around efficiency. So anything that reduces administrative burden will be a quick win. No one will argue with a tool to reduce paperwork. They will certainly push back against something that replaces their job. Regulatory is where I really like to focus. We see a lot of interest from our community around foundation models, like I said in my talk, around new approach methodologies or NAMs. The AI might not necessarily replace an experiment in the lab, but certainly it can prioritize a lot better than you can. So instead of doing 100 tests in the lab, you can do 1 billion tests on the computer. And that prioritization will limit what you actually have to do, which is really exciting. Over time, these models will just get better and the trust will evolve, and I think their role will increase. So we all need to sort of pay attention and start playing with them. Thanks, Liam.
Yeah, no, some really great points. Totally agree. Thanks so much, Daryl. All right. Good question here for you, Meredith. What else can AI tell us about an H&E image?
Yeah, it's a great question. So one of the big areas that I didn't touch on, but Daryl, I think you maybe teased very briefly when you touched on digital pathology, is the ability to predict. So things like genetic mutations, variants, or something like MSI status. This would fall in the bucket, right, of things that humans can't do right now. We cannot look at an H&E image and see if there are genetic mutations at play or, microsatellite instability. But there are AI models that are able to do that type of prediction. And depending on the indication and the biomarker it is that you're asking it to predict, the amount of accuracy can be so high that you don't even need to do confirmatory testing. Once kind of that initial pilot verified data, you've gotten through that step. Still have to qualify your tools, but the AI tools are so good in some spaces that once you've sufficiently qualified, you could skip testing completely. In others, it's just a way to, kind of stack the deck in your favor and only test ones that are much more likely to test positive or negative, depending on your desired outcome.
Yeah, and I totally agree with that. I think the triaging aspect, they're not necessarily always going to replace a sort of a doctor or a pathologist, but certainly triaging risk is something that they'll be very well suited to do in the near term. Which I really like. I also observe when we get a lot of requests is, the H&Es are great, but you have to pair them with appropriate metadata and real world data, or else you can't predict outcome. You can't predict treatment response. You can't do those. So we have hundreds of thousands of H&Es, but we have a much smaller data set that actually is usable because we don't have the outcome data or the biomarker data associated with it.
Completely agree.
Yeah, some excellent points, Meredith, Daryl. All right, Luke, we'll do one for you now. So what security measures are in place to ensure that my chats are kept private? I can speak from scientist.com's point of view. Like I was talking in my presentation, our new security, data security RFIs will go a long way to make sure that anybody on our platform is secure. But yeah, I'd love to hear from you, Luke.
Yeah, we get a lot of questions about this. People are concerned in our industry. Security is definitely top of mind when we built the platform. It's a web platform, so we use web standards, security practices, encryption in transit, and at rest we have an AWS cloud that we have encrypted block storage. So all the chats that people would have when they interact with the system would be stored encrypted. at rest, and nobody has access to those data sets or any of the logs except for specific engineers like myself. But otherwise, they're completely private. And then each individual user has their own user account that's tied to their email. And so you have a username, password, and the chats are not shared between users at all. There's a hard block at the database level to prevent any overlap. And we have other, security measures, like if you don't log out in a certain amount of time, your account will automatically log out, so on and so forth. The big the thing that most people ask us about is our use of a third-party large language model. So we work with OpenAI, and we do, by nature, have to send messages to their servers and get the response. But unlike, if you use ChatGPT, for example, they can basically see all of those messages, and they train on them, and they learn from them, and improve their model. We have a business enterprise agreement with OpenAI, and we use different API endpoints. And so the agreement is that none of the messages are stored, none of them are trained on. After we get the response, the messages disappear from their servers. So, they're, I think quite a trustworthy, in the scheme of potential LLM providers, they're quite a trustworthy one. And they've gone through, all kinds of security compliance checks. So we feel pretty good with them. I will say, in the future, if we start allowing people to bring their own compounds, obviously that will have to be completely hardened, and we would never send, let's say somebody's SMILE structure to any sort of third party. And we would, we'd release some papers on how we're doing that as we kind of build this thing out. But security is top of mind, for sure.
Yeah, fantastic. Great answer, Luke. All right, this next one, I think, Meredith, it's for you. Why would we expect tumor detect to give any answer different between run to run?
Yeah, great question. So the answer is, of course, the opposite. What we do expect from a tumor detect is to give it the exact same answer every single time. But and kind of like what you were just touching on in your last answer, Luke, right? AI learns when you give it access to more data. It continues to learn, continues to improve, and so that's maybe what you're getting at with this question is this AI learning and changing? And the answer is yes, but that learning and changing is locked internal and then will be released in, kind of named version releases. So like, think like an app update on your phone, you went from version 1.1 to 1.2, and those publicly available versions, that adaptive learning of AI tools won't happen. So the publicly released versions are very reliable, very repeatable, very reproducible, while still leveraging the power of AI to learn, adapt, change, improve for, then the next kind of named version release.
Excellent. Yeah, good
I
point.
could add, I have another follow-up on that. Meredith, did you see is there value in the heterogeneity of the H&Es that you actually collect and train on for these algorithms? I know we struggle with, we have, multiple blocks from same individual. You could take any one of those slices. It could be an edge. It could be, different cellularity, different content. So there's a huge amount of heterogeneity. Is there value in that heterogeneity for these algorithms? And is there a need potentially for reference data sets to help, sort of map that heterogeneity or diversity?
Yeah. No, I think tackling heterogeneity efficiently is really where AI is going to shine in the biospecimen space, because that's, of course, the biological reality. Tumors, in particular, thinking of oncology, are extremely heterogeneous, right? I gave the example of a mutation may only be present in 5% of cells, but that targeting that mutation could result, in an effective targeted therapy. So yes, heterogeneity absolutely matters. Training your AI models on a very vast heterogeneous data set. And what we're seeing is an increasing amount of both publicly available data sets, as well as, both of us are coming to the table with large banks of FFP samples, which means we have, large amounts of our own, kind of image repositories available.
Fantastic. All right, Luke, there's a nice question here for you. Can users adjust parameters like expression thresholds when querying mutation or gene expression profiles?
Yeah, it's a good question. So the whole kind of purpose of using generative AI rather than using just a traditional database web platform is that you can be as expressive as you want with the filters that you want to apply, the questions that you want to ask. So essentially, the large language model has access through tools to our database, like a kind of pared down version of SQL for people that are familiar with that query language. So it can basically write any sort of query of any complexity, and then you can use it as a chat. So if you ask for one thing and then you want to further filter whatever results come back, you can ask a follow-up question. So the whole point is to be as flexible and open-ended as possible, and get and not be tied down to kind of rigid filters that are predefined and that may not fit exactly what you are looking for.
Excellent. Yeah, great answer. Daryl, here's a question for you. What are your thoughts on generating or utilizing synthetic data in these data sets?
Oh, I love synthetic data. I think, it's one, because it's a technical challenge, and from a generative AI perspective, it's just a no-brainer that you would do it. I think the question is like, so how do you do it in a way that is sort of suitable? If right now there's this sort of feedback loop, which is a little bit problematic, where if we're synthetically generating huge amounts of data that go into the public domain, and then the AI models are then retraining on that synthetic data, it kind of creates a flywheel going in the wrong direction. So high quality data is incredibly important to pair with synthetic data. But if you want to do simulations or just prove out the edge cases, you typically have very rare things, and increasingly people want cohorts that are really challenging to sort of source. So synthetic data allows you to increase the numbers so that your sensitivity, your specificity, these edge cases can really go up. But there's a lot of benefits, but you have to know when and what to do it. Clinical trials is a great example. You could generate, generative AI around synthetic, clinical cohorts. There's huge things. So yeah, very powerful, but also quite a lot of potential traps.
Excellent. Yeah, some great points. All right, I think in the interest of time, we'll have one last question. I wish we had more time for the Q&A. There's lots of great other questions to answer, but we'll go with this one. So what new developments or research avenues are you most looking forward to over the next 5 years? Maybe Meredith, we'll start with you.
Yeah. So I know that we've been focused on preclinical research, but the thing I'm most excited about is actually the improvement to patient care. In particular, we talked earlier about how AI can predict that biomarker status from an H&E image, and I'm really excited for that to hopefully increase access to personalized medicine. Personalized medicine isn't a new idea. It's been around for a while, but paper after paper, even like just in the last week, JAMA published a new paper that only 35% of patients are getting the testing in order to determine whether they're eligible for these personalized therapies. And the personalized therapies are known to have better overall survival. So since H&Es are so common, they're already, kind of the backbone of standard of care pathology. It's easier to imagine us getting to a place where, doctors get a notification that John Doe's biopsy came back and he's very likely to test positive for X, so order the test. And hopefully that we'll see that 35% number of patients who are actually getting that testing and getting on targeted therapies go up.
Yeah, it's a really important point. Daryl, what about you?
I love what Meredith said also, because wouldn't it be an amazing world if we were sick, somebody told us we were sick, as opposed to we have to then go chase down a doctor and say something's wrong. So flipping the narrative. From a technology perspective, what I quite get excited about is the digital twin space around virtual cells and being able to do that. There's been some fantastic work that's come out of Chanzuk, as well as multiple labs and organizations about creating virtual cells that you can then do a host of in silico experiments on. So digital twins, virtual cells, I think those will be, they're going to be incredibly transformative over the next 5 to 10 years. I can't hear you, Liam.
Yeah, Liam, you're on mute.
Yeah, thanks so much, Daryl. No, I was saying, yeah, it's a great point as well. But last but not least, Luke, we'd love to hear from you.
Yeah, I think those are both great. I mean, I'm a software engineer, so I approach this more of like a tooling. And on the Gen AI side, I mean, I think we have these foundational generative AI models that are quite good at the moment, but what we're really lacking is the tooling behind it, connecting it to our common workflows in the preclinical world, as well as in every other part of the economy. But essentially, creating the tooling to connect it to these workflows, to connect them together. And once we have these agentic systems, I think we're already seeing it, but I just think there's going to be a huge proliferation of companies being started with a lot less capital, a lot less human overhead required to get up and running, because you'll have these systems that are able to really empower one person to do what previously a team needed to do, right? And all these things. So you can imagine, like, there has to be so many researchers, PhD students that have these great ideas, but they're dissuaded into starting companies because of the activation energy required. And I just think if that all comes down, we're going to see this huge proliferation. So I take the kind of optimistic view where some people say, oh, we're going to lose jobs. Yes, that might happen, but I think we'll also see this proliferation of new oncomers into our space with new ideas that may be more risky, but you can afford to have more risky ideas when the capital required is so much less to test those ideas. So I'm really excited to see what happens in our space over the next 5 years.
Yeah, it's a really interesting point. I love that. All right. Well, thanks so much to the three of you for being here and sharing your expertise with us. It was really a pleasure having you here.
Yeah, likewise. We really enjoyed it.
Thanks, everybody.
Thanks, everyone.
Yeah, definitely. Big thanks to the audience for joining us today. In closing, we hope you enjoyed the Scientist.com, Discovery Life Sciences, Bio-IVT, and Certus Oncology webinar. And I hope you enjoy the rest of your day. See you next time, everyone.